Can you see the slide? Yes?
Yes.
Okay. Alrighty, so today I'm going to be talking about next generation sequencing, and our lab at the National Omics Center, we work a lot with genomics and transcriptomics, and we use a lot of next generation sequencing technology in our work. And so I would like to share with you the sort of applications of these technology that could be used in the real world. And we work mostly with plants, so a lot of examples from the lecture will be on plant. But we do have a lot of animals and coral stuff towards the end of the talk. So before I start, I would like to give all these people credit for doing all the work. So this is people at the National Omics Center at NSTDA. We used to be with biotech, but now we split off and become a new unit. And so our center covers genomics, transcriptomics, proteomics, and metabolomics stuffs. Alrighty. And so the main reason we do all the stuff we do here, especially with plant, trying to improve the varieties, is because, as you all know, the population is growing, right? And even though the birth rate is coming down, there's still more people being born into the world every day. And in the next 30 years or so, there won't be enough food for everybody. So the way we look at it is that right now, all the land that could be used for growing food, it's pretty much used up. And so the more people we have, the less land per capita we have each year. And so there's also a problem of climate change. And so the amount of food that we can produce each year is not enough to feed the world. And so in order to increase food supply, there are a couple of things you could do. You could use modern fertilization, you could use pesticide, or you could do precision farming, but all of those takes technology. But one of the things you could also do is to improve the varieties themselves, to make sure that the plants that you're growing have better quality, have higher yield. And so what I meant for when I said better varieties is the varieties that are resistant to disease, resistant to bugs. They are resistant to strange conditions that we face, like drought or salinity or flood. And also, we also want fruit and vegetable and crops that have higher nutritious value, or food that tastes better. So there's also breeding for tomato that taste better, that are bigger, that have good textures. And so all of these can be improved through a traditional way, or you can use DNA or genetics to help. And so before I dig into the details, I would like to give you an overall research directions that the National Omics Centers are involved with. And so we are doing two main sort of topics. One of them is agriculture, and the other one it's involved with biodiversity and bioresource and more environmental stuff. And so for the agriculture, we focus our energy on marker-assisted selection, which is the use of DNA marker to help improve the varieties of crops. And the crops that we work with are mainly economic crops like rubber tree, sugarcane, corn, oil palm, and also seed vegetables. We also have biodiversity projects, which are really cool. Actually, they're somewhat random, but they're kind of cool. Like the SNP identification project, with the tigers, the DNA barcoding project for our species identification with the herbal plant species, and also the coral projects looking at diversity of corals in Thailand, as well as the diversity of the bacteria that lives with the corals. And so I will start with the agriculture, and then we'll move on to the biodiversity side. But before that, I just want to give you a background of what I did for my PhD, which has nothing to do with what I'm doing right now. And so when I did my PhD, I worked with a little green alga called Chlamydomonas, and it's a single cell algae that it's a progenitor of land plant. And so we use it as a model species for studying plant. And what I looked at when I did my PhD is I study how these algae responds to sulfur deprivation. And so this is when algae is like dying because it doesn't have food, it doesn't have sulfur. And then when you spray with some sort of substrate, you can see that the algae is making an enzyme that can cleave sulfate off of material so that it's a survival skill for algae. And that's what I was looking at during my PhD, which is over 10 years ago. And then when I graduated, I ended up working with something totally different. And my first project was actually on oil palm. And if you remember, about 10 years ago, oil price was very, very high. And Thai government had a lot of budget for oil palm research because we use palm oil for biodiesel. And so these are the people who helped me with the oil palm project. And the reason why we are interested in oil palm is because it's actually the most efficient oil-bearing species in the world. Like a lot of people know that soybean produces a lot of oils, sunflower, rapeseed. There are a bunch of temperate species that produce oil, but none of them compared to oil palm. If you look at the amount of land grown for each species, you can see that soybean took up a lot of area. Oil palm took up only 4% of the growing area, but it accounts for a third of the total oil production in the world. And so it's very efficient, like photosynthetically. And that's why we focus on oil palm. And the other thing about oil palm is that it can only be grown in the equator area. So Thailand, Malaysia, Indonesia. And so if you're a plantation owner, what would you like? What kind of trait would you like with oil palm? And one of the things is that, of course, you want it to make a lot of oil. And for oil palm, I'm not sure if you're familiar with the fruit of oil palm, it looks like this. There are three fruit types. So there's the dura, the piscifera, and the tenera. So the dura, has a lot of thick shell, which is not desirable. The piscifera is the desirable one because it's got a lot of oil. So the orange area is where you can get oil. But it's sterile, and so you can't make progeny out of this. And so when you mix these two, you ended up with the perfect progeny, which has thin shell, but it's also fertile, so you can continue making progeny. And so Malaysia has sequenced the oil palm genome, and then they figure out the gene that control the thickness of the shell. And it's a single gene, and it's a dominant trait. And so if you have a dominant allele, you get thick shell. If you have recessive allele, you get no shell. And when you mix them, you get a heterozygous that has a perfect thin shell. This is what you want. And so the other thing that you would like in oil palm is that you would like the tree that will give you more female than male flowers. And so, you can grow oil palm like in the northeastern area right now, but the problem with that is that it only gives you male flower like this, which is pretty much useless because this doesn't give you any oil. The only way you get oil is when you get female flowers, which give you the fruit branches like this. And so what we want is oil palm that only flowers the female. And so you can select for the palm tree that has a genetic background that has a high female to male ratio. And this is fortunately controlled by a gene as well. And so there's a way to select for it. But I didn't work on this. My first project was actually to get oil palm that is short, kind of like short coconut tree. And the reason you want short trunk is because when you have short tree, it's very easy to harvest the fruit, and you could grow them for much longer. So the economic lifespan, it's like 20, 30 years, and there's less damage to the fruit bunch. And so we would like to get a tree like this. And when it's tall, you have to cut it down after like 10, maybe 10, 15 years. So the problem with oil palm is that it takes forever to breed. This is the conventional breeding. It takes 10, maybe 12 years to go through one cycle. And to get a good variety, you need maybe 3 or 4 cycles, and that's 40 years. If you replace traditional breeding with the marker-assisted selection, it could be a lot quicker. And so you can only need to grow it for maybe a year and a half, and then you extract the DNA from the leaves and see if it has the trait that you would like or not. And you don't have to waste your time with the phenotypic evaluation. And so the breeding cycle would be shortened by half at least if you use DNA markers. But before we can talk about marker-assisted selection, I would like to introduce the molecular markers just in case some of you are not familiar with DNA markers. And so DNA markers are pretty much any marker or any DNA position that it's on the genome. So let's say the yellow line is the whole genome, and then you have DNA marker like spread throughout the genome. There are several types of DNA markers. You might have heard about the simple sequence repeat or the microsatellite. These are the marker that are simple sequence repeat like TA, TA, TA. And the allelic difference would be the number of repeats in each allele. And so the top allele has 3 repeats and the bottom one has 5. This type of marker was actually very popular maybe 10, 20 years ago, but nowadays they're being replaced by single nucleotide polymorphism or SNPs. And the SNPs are just a single nucleotide change. So for example, a change from T to C here, it's a SNP. And the number of SNPs in the genome are a lot more prevalent than the SSR. So if you use SNPs, they're pretty much everywhere in the genome. And so the reason you want to use DNA markers is because if you have a certain trait of interest, like fungal resistant or genes that control yields or gene that control drought resistant, these genes are usually next to some DNA markers. So for example, this gene is close to these 2 markers, the gene controlling yields close to these 2, and the gene controlling drought resistance are close to these 2 markers. And so these markers are useful for selecting plants that have the trait of interest. But given that there are markers everywhere and some of them are bound to be close to the gene of interest, you have to figure out a way of identifying which markers are close to which genes. And so the way we identify the linkage or the association between DNA markers and the trait that you're interested in, genetically, you have to generate a segregating population. And basically what a segregating population is that you cross 2 parents that have very, very different phenotypes. Like in this case, if you're interested in disease resistance, you would cross a parent that is resistant with a parent that is susceptible. And then the progeny will segregate. And so you will get some progeny that are resistant, some of them are susceptible. And then you genotype all of the progeny, and then you phenotype them as well. And so you test to see whether they're resistant or they're susceptible, and then you extract the DNA from these progeny and sequence the DNA to get the SNP markers. And when you get the phenotype here on the left-hand side, and so you get some plants that are resistant, you get some that are susceptible, and then you genotype each one of the progeny. So progeny number 1, number 2, 3, 4, 5 to get a SNP marker. And then you can look through these. I mean, technically you don't actually look through it with your own eyes. You have a software that will do this, but I simplify the table. And maybe you could try to look at it and see which marker is linked to the trait of interest just by scanning the table. Does anyone want to take a guess? No? Yes?
It's still very difficult to guess. Is there a way to guess from this?
Usually,
Takes
like
time.
yeah, it takes time to look at the table. And so if you look at marker number 9, you will see that one of the alleles, it's linked to the resistant phenotype, and the other allele is linked to the sensitive phenotype. And so if you have a plant that has a C, it's likely to be resistant. And if you have a plant that has an A at this SNP position, it's likely to be susceptible. So this is how you guess, how you predict whether or not a plant of unknown phenotype, if you have the DNA, you can predict whether or not the plant would be resistant or susceptible. In reality, you would have a lot more plant. Like I only show 8, but in reality, you would get 100 or 200 or even like 1,000 plants. And you also have a lot of SNP markers, like thousands of SNP markers, in order to identify which one is associated with the trait. But this is the basic idea. The more SNPs you have, the higher the chances that you will find the marker of interest. And so if you look at these, on the left, it's the genetic map that is built with the SSR or microsatellite marker. You can see that it's not a very dense genetic map compared to the genetic map on the right that is built with SNP markers. And so when you identify the location of the gene that's responsible for the trait of interest, which is represented by the star, you can see that the closest marker to the star is actually very far for the microsatellite map, but it's very close with the SNPs map. And so it allows you, the SNP allows you to get really close to the gene that you're interested in. And so when you're doing a selection, these will get you the plant that has the correct phenotype. Like the probability of getting it correct with the SNP marker will be higher than the microsatellite marker. And so how do you identify SNPs? And so we use next generation sequencing to sequence a lot, just sequence basically the same spot in the genome. And then we do a sequence alignment. And so you take the read and then you align it with the reference sequence. And you can see that there are certain positions that there are the polymorphism in the nucleic acid change or nucleotide polymorphism. So like this is the SNP position. So the accuracy of identifying SNPs depend on the read depth, which means that if you only have 3 reads here, you might not see that this is the SNP position. But when you sequence more, you would realize that at this particular locus, there's T allele and there's also a G allele. And so it makes this position as SNPs. And so in order to accurately identify SNPs, you need enough read depth. And because plant genomes are pretty big, even though the sequencing cost is like really cheap these days, if you have to sequence the entire genome like multiple times in order to get the read depth coverage that you want, it'd still be pretty expensive. And so the way we do the experiment is that we only sequence a certain portion of the genome. So we use restriction enzymes to cut and select only a certain portion. And so we reduce the sequencing cost by doing this. And the approach that we use to reduce genome complexity, or the approach that we use to sequence only a certain portion of the genome, is called genotyping by sequencing. This technique uses restriction enzyme to digest the genome. And so you only digest and get this fragment, and you sequence just the fragment like over and over. And so you get a lot of read depth that will allow you to identify the SNPs. So this is the way you do the GBS. You take the genomic DNA and then you digest it into a small fragment. And then you put the adapter, you put a sequencing adapter on it. And then you do a PCR amplification to get a lot of fragment and sequence that. And once you get the sequence data, you align them back to the reference sequence, which is the yellow line. This is the reference sequence. And then you align the short read back to the reference sequence and identify the SNP's position like this. And so if you sequence it this way, you will save about 100 times, because if you have to sequence the entire genome, it might cost you $1 million baht, while this approach will only cost you 10,000. You guys sleepy yet? So let's get back to the original question of when we asked what the plantation owner would like with their oil palm. And so what they want is a short palm, because it's easier to harvest. And so in order to identify the genes that are associated with short trunk, we have to cross between two parents. And so this is the female parent that is tall and the male parent that's short. When you cross them, you get an F1 that it self-fertilized to give you an F2 progeny that segregate. And so the F2 progeny would have a really, really short tree, as well as a really, really tall, and anything in between. And so we use this population for genotyping and phenotyping. So the first thing is that you would identify SNP markers in the segregating population. So it's the F2 population, this one that I just showed you. So we'll find the SNP genotype and then construct a genetic linkage map from those SNPs. And after that, we can use the genetic map to locate the QTL, or the gene that controls the height. And so, but in real world, it's not this simple. Like, it's not like you set up a cross and then you genotype the progeny and then you get the linkage map. During this project, it turns out that we don't have the tree from the dad. So this tree died a long time ago. And so I was able to collect sample from the mom, the F1 and the F2 progeny. And so it creates a little bit of obstacle, but we can get around it. And so when you do the sequencing of the progeny, you will realize that some of the SNPs cannot be used. So when you don't have the male progeny or the male parent, the only SNPs you can use is the SNPs that are heterozygous in the F1. And so if the F1 is an AG and the parent is an AA, you can assume that the F1 received the G allele from the dad. So we're going to select those configuration of SNPs and use them. So on the next slide, so we genotype the F2 progeny and we get roughly 13,000 SNPs. And from those 13,000 SNPs, there are only 3,400 SNPs that are heterozygous in the F1 and homozygous in clone A. So these are the type that we can use because we can guess which allele comes from the dad. And so we take 3,400 SNPs and build a genetic linkage map, and this is what you get. So you get a map that is really dense. And when you combine this map with the phenotypes, you can see that there's a QTL, or the genes that controls height in oil palm, on chromosome 14 and chromosome 15. And these are the positions of those genes. Once you have these genes, what you could do next is try to develop a marker. Or first thing we did was to try to see if these genes are actually close to any proteins or genes that we know of. And so this is the region where we found the QTL. And it turns out that it's next to two proteins, the DELA protein and the GA2 oxidase. And both of these genes have been published earlier, and they are involved in gibberellin homeostasis. And gibberellin, it's a plant hormone that controls height. And so we know that we get pretty much the correct gene for the QTL, because we're studying trunk height. And so from this step, we decided that we would like to develop some sort of SNP assay for selecting oil palm. So we take the sequence from linkage group 14 from the genes or from SNP marker that are linked to the height from linkage group 14 and 15. And for this particular SNP, we know that a genotype AA will give you short plant. So the absence of T allele will mean the plant will be short. On the other hand, for the second SNP, if you have a TT, the plant will be tall. And so the absence of C allele makes the plant tall. And so when you have these two markers, we can use these to select or to predict if the palm's going to be tall or short. And so we develop a TACMAN assay for genotyping 85 or actually 89 oil palm. And so among these 89 palm trees, there are 51 short, 38 tall tree. And the genotype of each short and talls are in the table. And so if you genotype with the first SNP, we already know that the absence of T allele means that the palm are likely to be short. And so we look at the AA, and all the AA should be short, right? So what you do is that you keep all the AA because that's what you want. And then you genotype it again. With the second marker, the absence of C allele means it's tall. And so the TT is tall. So you throw away the ones with the TT and you end up with all the short plant. And this is how you use the DNA marker to predict whether or not the palm will be short or tall. And you can do this when the palm is 1 year old. You don't have to wait like 10 years to see that, oh, it's a tall palm, we have to cut it down. And so these markers are being validated in the field, and hopefully we can use it. We already patent these two markers. All right. So just to summarize what we've gone through. And so the question is, the farmer wants a short palm tree. We develop a GBS technology to identify SNP markers that link to the trait. And then once we identify the marker, we validated it. And these markers are going to be used in breeding program in order to get short palm in a short period of time. You don't have to wait 10 years. So if you'd like to read more about the stories, you can read it. We published it a couple of years ago, so you can look at these papers and see how we did the linkage map and QTL analysis. All right. Any questions on this? Oil palm and linkage map and SNP markers? No? Okay. Not sleeping, right? You guys are still. So before we move on to rubber tree, I just want to quickly show you.
Yeah. Are there any question? I think before Ajahn go to the next story, you might forget what you want to know to ask, right? So maybe you can take this time. Some part that maybe is a little bit fast, you cannot follow or whatever, you can also ask as well.
Sorry, Ajahn, I have a question. Ajahn.
Yes.
Did somebody ask a question? Sorry, I didn't hear.
First of all, Ajahn, it's very well. I mean, very nice presentation and also a lot of information that many of the points I got like some little bit, but it's really very interesting and also very nice presentation. But Ajahn, question about is farmers that want the short palm tree, right? The question is right, and then they develop the GB that they develop the method for SNP. But my question is, why the farmer want the short palm tree? I mean, is there any drawbacks? I mean, any consequences of the short palm trees? Thank you.
Why do the farmers want the short tree? Is that your question?
Yeah, Ajahn, I mean, why the farmer want short palm trees, and is there any drawbacks, like any consequences of this tree?
Oh, they want the short tree because it lasts longer. So you can grow them for 30 years. The tall one, you have to cut it down after maybe 15 years. The short tree is also, it's also easier to harvest from the short tree. Because, you know, imagine like trying to bring down the big like fruit bunch from a really tall tree. It's very difficult. And in Thailand, we don't use mechanical harvesting. We use like human to harvest oil palm. So it's obviously easier to harvest from the shorter tree, and that's why they want short tree mainly. Yeah.
All right. So, Ajahn, is there any drawbacks, like any consequences?
No, actually, no. Yeah. We try to breed for a short tree that still have higher yield. Yeah.
Okay. Thank you.
You're welcome. Alrighty, and so before we move on, I just want to give you a quick comparison of the SNP discovery approach. And so today we talk mainly about the GBS approach, but there are also, you might have heard of RNA sequencing, targeted enrichment sequencing, or the shotgun whole genome sequencing. And so just to give you a quick comparison, GBS, it's the most cost effective, it's quick, and you don't require any prior knowledge on the genome. And you also get SNP everywhere from the genome. With the RNA-seq, you only get SNPs from the gene. With targeted enrichment, you only get SNPs from your target. And with whole genome shotgun sequencing, it's pretty much the same as GBS, but it's a lot more expensive. All right, so we can also apply GBS to solve another problem, not just the short trunk, not just the oil palm. We applied it to develop the disease-resistant rubber tree. And so you're probably aware of rubber tree. I mean, if you live in this area, it produces latex that have really, really cool property, like it's very heat resistant, it's very resilient. And so airplane tire, they have to be made from natural rubber tree. They can't be made from synthetic rubber because synthetic rubber are just not as superior as the natural rubber. The natural rubbers are a very complex, large molecule, and they can withstand a lot of heat, especially during landing of the plane. And so it's very important that we produce this rubber tree. And the reason for that is because rubber tree originates from the Amazon area in South America. But if you look at the production of rubber tree, it focuses mainly in Thailand, Indonesia, Malaysia, and none from South America. And the main reason is because there was an attack of a fungus back in the 1950s. It's called South American Leaf Blight. And it wiped out all the rubber tree plantation in South America. And so we're still pretty lucky. The disease hasn't made it to Southeast Asia yet, and so we can still grow rubber tree. But it's a very dangerous disease, and it's actually considered a bio-war material, this particular fungus, because it can wipe out all the rubber tree plantation on Earth. And even though we don't have the South American leaf blight, we do have two fungus or fungal diseases in Thailand caused by Corynespora and Phytophthora. And these two can kill rubber tree in Thailand. And that's why we would like to breed for a resistant variety or resistant cultivar. And in order to identify, so first we have to identify which genes or which marker associated with the disease resistant. We generated two populations. We cross the resistant with the sensitive, and we get the segregating population. So the segregating population looks something like this. So we genotype these. So we know all the SNP marker genotype of each of the 79 F1 progeny. We also phenotype it so we know whether or not it's resistant or sensitive. And we do that for both populations, and we build the genetic map. And so this is the statistic for the genetic map. So each map contains a large number of markers. But what you can do, because if you remember, if you notice, both of these populations share the mother. So BPM24 is the mother for both crosses, and so that allows you to combine the map. And so we can merge these two maps, make the consensus genetic map, which has even more marker. And so the distance between the marker is narrower, which means it has higher resolution. So if you look at the picture, it might be easier to understand. And so the consensus map is the blue one in the middle here. Each individual map is the green and the pink. So you can combine the information from the green and the pink and generate the blue map, which is really, really high resolution. And we can use the consensus blue map to do the mapping. So this is the analysis with the Corynespora resistant. And you can see that there are SNPs on linkage group 17 that has association with Corynespora resistant. And so we are repeating this experiment. So we have to do more phenotypic characterization because this data is only based on one replicate. And usually you do 3 replicates. And so right now the Rubber Institute of Thailand are collecting more phenotypes so we can confirm whether this position is really linked to the resistant gene. And if it is, we can do the similar experiment with oil palm where we design the SNP assay and then test whether or not it can predict the resistant in rubber tree. And if it can predict the resistant, then we can use this in breeding programs. So if you like to read more about how we construct the genetic map in rubber tree and how we can combine 2 different maps into one to generate a high density map, you can read it in this paper. Okay. Do you guys need coffee? Need a break? Are you sleeping yet? Okay. So besides the application of GBS for breeding program, disease resistant, tall tree, short tree, or high yield, low yield, the use of SNP marker, it's not limited to just that. We can also use it for something else. For example, cultivar identification or seed purity test. And so just to give you an example, for a company that, for seeds company, there are different varieties of cucumbers. And if you look at the fruit, you can sort of tell the difference. But if you look at the cucumber trees or the bush or the plant, you can't really tell them apart. And they have a lot of difficulty differentiating one variety to the other. And they don't want to wait until it gives you fruit. And so if you want to know, like when the plant is really young, if they will give you this type of fruit or this type of fruit, you can check the DNA. And so the way to do that is by identifying SNPs that are specific to each varieties, and you can use those SNPs to identify each cultivar. Besides the cultivar identification, you can also use SNPs to do seed purity testing. And so I'm sure you're familiar with these. You can buy them in supermarket and stuff. These are F1 seed, which means that they are produced from a mom and a dad that has really, really good quality. So this is the example. Like when you buy a melon in the supermarket, right? These melon are produced from the hybrid seeds. They're both sweet and disease resistant. And they were crossed from a mom that is sweet and a dad that is disease resistant. But you know, sometimes when they cross these two parents, there will be contaminated pollen coming from somewhere else, and that will result in contaminating seed. And so the contaminated seed will not have the disease-resistant father. It will have some random dad. And when you take this seed and germinate it, it will give you a melon that's rotten because it's not disease resistant. And this type of melon is actually really, really bad for the reputation of the company, because when they sell you the F1 seed, they promise you that the melon you get will be sweet and disease resistant. And so the company will have to test and make sure that the seeds come from the correct dad. And the way they're doing it right now is by doing what's called a grow out test. And so you also have to check the purity of seeds like corn. So if it's not coming from the correct dad, it could be ugly like this. And so this is how they test for the purity or the paternity test for seeds. And so they basically grow them out in the field. And then they send people to look at each plant to see if they look like what they're supposed to look like. And you can imagine this takes forever. And it's not very accurate because, you know, people make mistakes all the time. And so not only does it take 6 months or 12 months, it's also not accurate comparing to the DNA test. And so if you use the DNA to test whether or not it comes from the correct dad, it's very quick and it's very accurate because the DNA doesn't lie, and you can do it within 2 days instead of like 12 months. And that's why we developed, so the National Omics Center developed a SNP-based test to see if the F1 comes from the correct parent. And so we also offer the seed purity testing to seed production company. And not only would this technology save the seed production company some time and some money, it also helped the farmer produces more, because instead of having to wait like 6 months or 12 months for the test result, the DNA-based technology is very quick. And so when you know the result quickly, you get paid quickly, you can invest that same amount of money into the next crop season. And so in a year, you can grow 4 instead of just 2 crop seasons. And that means you make a lot more seeds. And this is also good for the economy of the country. So the seed production company gets to produce more seed, the farmers get paid much quicker. And so, and they can also grow 4 instead of 2 crops, and they make a lot more money. And Thailand can also export a lot more because there are a lot more seeds produced. And so this is a win-win situation for everybody using DNA tests. All right, and so you guys know, like, it's really hot these days. And so my cat started doing like really strange stuff. Every time we open the fridge, they would jump in like right away. And sometimes I would leave, like, I didn't leave, I sort of forgot and like left the cat in there for like half an hour, and he's like actually really happy. And so when I told him that, okay, you have to come out because we're gonna close the fridge, one of them like pretend to be an orange juice carton and like, no, don't take me out. Okay, just to make sure you guys are not sleepy, showing you some cat pictures. All right, so we're gonna switch from plant to biodiversity. So maybe I don't know if you guys want to take a 5-minute break, a bathroom break, go to the bathroom, get some drink, and then maybe we can meet back here in 5 minutes at 5:20. Does that sound? Is that okay with you?
Okay, ka.
Okay, let's take a 5-minute break. You guys could go to the bathroom or get a drink or something, and then we'll meet back here in 5 minutes. All right.
Okay, ka. Take a break ก็โอเคจ้ะ
Hi. Is everybody back? Can we continue?
Yes, yes, sir.
Okay, ha. Alrighty. Okay, so the first section would I talked about it's mostly on agriculture, how you use SNP markers identified through NGS technology to do marker assisted selection, to do the breeding program, like to expedite breeding programs, how you could get varieties that have trait of interest like resistant to certain fungus or to certain bacteria, and high yield, short tree, blah blah. The second section would be a little bit different because we're going to switch gear and talk about biodiversity and bioresources. This will be more on the environmental side and somewhat of a random subject. And so a lot of these works are actually collaborative projects with other governmental units or universities. And so the first project I would like to talk about is the coral bleaching project, which this is our collaboration with the Department of Marine Science, no, the Marine and Natural oh my God, I forgot the name of our collaborator, but anyway, they're the department that take care of the natural resources, especially the marine. And so
Very beautiful. Where is this?
I took it from the Times magazine, so I actually don't know. But
Okay.
a lot of people said that the white ones are beautiful. So no, the white ones are the dead one. Okay, by the way.
Dead, right?
Yes.
Yeah, but they still look like very beautiful. Like I mean,
Yeah.
a lot of coral.
Yes, and so
Yeah.
that's why they said it's a most beautiful death, but it's
Oh,
actually
that
very
was beautiful. Yeah.
in 2016, 5 years ago, we have a big coral bleaching problem in Thailand, you know, in the Andamans and also Gulf of Thailand. And if you're not familiar with coral bleaching, this is what they do. They look like they are completely white. And so on the left side, this is what coral is supposed to be. They're supposed to be brown and they're supposed to have a lot of fish. It's a big ecosystem in the marine environment. But when the temperature goes up, when the water temperature goes up, something happened, and we are not sure yet actually. And so if I zoom in, this is the corals on the left. This is the coral with the algae living inside, and so they're living in symbiosis. These algae can do photosynthesis and share some of the carbon with the corals. The coral would utilize the carbon to build the skeleton. So all the calcium carbonate that you see, it's actually the energy that is used to build this. It's from mostly from the algae, and the coral provide like shelter, nitrogen, amino acid to the algae, and so they live together happily until the water temperature increases, and that's when the problem occurs. And so the scientists are not sure whether or not the algae like couldn't stand the coral and leave, or the coral host kick out the algae. Because when the temperature goes up, there might be some sort of production of reactive oxygen species, which are not good. And the corals might not be happy, and so they kick the algae out, even though they know that they're gonna die without the algae. And so this is coral bleaching. So they bleach out of color because the brown color comes from the algae. And so the Department of Natural Resources, they have a program called Coral Reef Restoration Program. That's where they take the corals from the sea and then they grow them in nursery like this. And then they take these corals and transplant them back to the natural reef site. But the problem is, next time the temperature goes up, these transplanted coral ended up dying again. And so they wanted the corals that are heat resistant. And so let me show you this video. If you notice, there are some coral that bleach, but there are also some coral that remains brown, right? Not everybody bleaches, which means that there are certain genetic difference between these two corals. They're the same species, you know, but there are some sort of genetic difference that allow one of them to grow normally and the other one bleach and die. And so our original goal was to identify DNA markers that are associated with heat tolerance. So when I started this project, I did some basic, you know, just Google search. I just want to see what people have done in Thailand. And it turns out that there was absolutely nothing known about corals in Thailand at the molecular level, meaning that a lot of work done in Thailand was just the survey. They survey if the coral dies, if the coral leaves, but there's no information on the genetic material. There's no information on the genetic diversity. Pretty much there was nothing known at the molecular level. So it's like a blank space here. So I decided to go back and started with looking at genetic diversity of corals, because the genetic diversity, it's an important indicator of whether or not a species will become extinct in a, in a short period of time or not. And so in order to look at the genetic diversity, I use the fragment that is called internal transcribed sequence, or ITS, ITS1 and ITS2. It's the sequence between the ribosomal RNA. And so the ribosomal RNA sequence itself is highly conserved, but the internal transcribed sequence they mutate naturally, and so you can use this sequence as a marker for telling how diverse the member of the species are. And so I use the ITS for corals and for the algae. The algae is called Symbiodinium. And I use 16 srRNA sequence to evaluate bacterial diversity. And so we collected samples from different places in Thailand, both on the Andaman side and also from the Gulf of Thailand. And Dr. Lalita, or Dr. Nan, it's my collaborator from the Department of Natural Resources, and she was the one doing all the coral sample collection, which you might think it's fun, but it's not very easy at all. If you look at her, she has to tie a lot of bags while like scuba dive to collect the samples. And I will show you a video of her collecting coral sample, and you will realize how difficult it is. It's actually extremely hard to get the samples into the tube in water. So this is on a good day. On a bad day, there would be a lot of wind and the wave is really strong. And once you scrape out the coral sample, it just floats away and it won't go into the tube. And so she spent several months collecting samples for me, and I'm actually very grateful for that. She's an awesome collaborator. So once we have the sample, we extract the DNA and then we do the PCR. And so we amplify this fragment from ITS1 to ITS4. These are the two primers we use. And we use a technology called PacBio to do a sequencing. And so PacBio is a sequencing technology that could get a sequence of a really long sequence from a single template. And so I'm not sure how familiar you are with PacBio, but let me show you how the technology works, because I think it's a really cool technology. And so this is the PacBio chip on the bottom left. And so within this chip, there are many, like several thousand, actually a million holes or wells. So each of the well looks like this on the right. So this is a single well. The DNA is attached to the bottom of the well with the polymerase. And when the DNA is sequenced, the nucleotide or the DNTPs are labeled with fluorescent dye of different colors. And so there are 4 nucleotides, there are 4 different colors, right? And each time a single nucleotide is incorporated, it will flash a certain color of light. And so each nucleotide has one color. And when the DNA is sequenced, there's going to be a camera that records the color that it actually came out. And so you can look at the color and then translate that into the sequence. This is how PacBio works. And you can get a sequence that is very, very long. Some of the sequence can be 50,000 base pair, extremely long, very cool technology. And so this technology could be used to sequence full-length amplicon like 16 srRNA, like the ITS, or any gene of interest. You can have a 5 kb gene or a 10 kb gene, and it can be sequenced in a PacBio. So if you compare PacBio to current sequence technology or to sequencing technology that has been developed Sanger sequencing is the oldest one, and it's very, very low throughput. Illumina or MGI has very, very high throughput, but at the same time, it gives you a super short sequence, 200 to 500 base pair. PacBio give you a medium throughput, but the read length is very long. And so you can choose different sequencing technology based on what you really want. Like if you don't care about the read length, but if you want something cheap and you want a lot of data, go with Illumina or MGI. But if you want a really long read and you can afford to pay for PacBio, which is more expensive, of course, then go with the PacBio. So, like I said, PacBio has the advantage of giving you a long read, so you can read the full-length sequences. If you use the Illumina or MGI, you can read just the V3, V4, or the V5, V6. But with the PacBio, you get the whole 16S rRNA. And the way PacBio works with the amplicon sequencing is when you have the PCR, right? You would generate a library by ligating a SmartBell adapter. And the SmartBell adapter is a single strand that is a hairpin, right? You ligate it on a double-stranded DNA. And when you ligate it, you ended up with one big circle that is a single strand template. And the single strand circular template is going to be sequenced with the DNA polymerase. Remember that well at the bottom of the well, there's a DNA polymerase? And the DNA polymerase will sequence these over and over and over because it's circular, right? The DNA will just keep going in circle. And it will keep going until the DNA polymerase get really tired and then fall off. And you get a really long sequence that looks like this. So it sequenced this blue fragment, red, blue, red, blue, red, blue, and then keeps going. Once you get this sequence, you trim out the red sequence because it's the adapter, you don't want it. And then you get the blue fragments and then you align them and do some correction. And you ended up with a read that is very, very accurate, almost as accurate as Illumina read, but you get a really long read instead of a short one. So, and this is the raw data. This is the sort of data you actually get. And so when you sequence it, you can see that the PacBio goes like 20 kb, 40 kb, 60 kb. And after you take the consensus sequence read, the 16S rRNA gene is 15,000. No, it's 1,500 bases. And so this is the PCR product or the amplicon that you like to sequence. And one PacBio cell can sequence like roughly 50,000 or 25,000 amplicons or different DNA molecules. So if you would like more, you have to use more cell. This is with the first generation PacBio. The current generation, I believe this number goes up to like millions already. And so you get a lot of throughput with PacBio as well. So the reason we sequence the ITS is because we want to look at the diversity of the corals. And if you compare the Andaman Sea and the Gulf of Thailand, you can see that the distribution of corals in the Gulf of Thailand, it's a lot better compared to the Andaman Sea. So on the Andaman side, there is one particular type or one particular genotype of corals that takes over, and a lot of genotypes are becoming extinct, disappearing, which is not good. And if you look at, so this is the summary of the whole, all the collection sites on the Andaman Sea and all of the collection sites on the Gulf of Thailand. A more detailed distribution is in this picture. And so you can see that Manai is good, Matra, Rangkajil, this has a good distribution, Gatao as well. The area that has problem, it's like Payu, it's like being dominated by a single genotype, and the rest of them are becoming extinct. Same with this. So two main genotypes, the rest are becoming extinct, and this is not good. And so this type of data is useful for the Department of Natural Resources. They know which area they have to take care of in particular. So Payu would need a lot of attention, Ngam Yai, and also Sing Island. And the rest of them are still pretty good. We also look at the algae distribution. And surprisingly, the algae distribution looks the same on both sides, Andaman and the Gulf of Thailand. And this is the comparison between the diversity of the coral host and the algae that lives with the corals and the bacteria that lives with the coral. Overall, I would say that Gulf of Thailand is doing pretty well. Andaman Sea has a little bit of problem with the diversity. We also look at bacteria that lives with coral during coral bleaching. And so we did the study before bleaching, during the bleaching, and after the bleaching. And so before it's in March, the temperature is 28. During the bleaching, temperature goes up 3 degrees, and then the temperature came back down. And so we look at what sort of bacterial diversity is associated with each of the condition of corals. And so you can see that a lot of gamma proteobacteria are associated with corals when the temperature is down. When the temperature is up, alpha proteobacteria takes over. And we notice that some of the bacteria that causes disease, like pathogens, are actually alpha proteobacteria. And so we hypothesize that when the coral bleach, they're weak. And so some of the opportunistic bacteria started to take over. But when the temperature is back to normal, the corals is healthier. And so the gamma proteobacteria come back. I mean, these sort of studies are mostly for basic science. They generate knowledge. If you ask me what you could do with this data, or what you could apply it to, as of now, it's very difficult to answer that. But you can sort of use the bacteria as an indicator of whether or not the coral is healthy or not doing so well. So we also did another experiment looking at the bacteria in a controlled environment. And so this is from the ocean, which has a lot of variability. There's so many factors involved. And so we set up a similar experiment in the tank and look at the bacteria. And we found that it's very similar. So when you're under the control environment, the temperature is down, you get a lot of gamma proteobacteria, and you get a lot of alpha proteobacteria when coral is under stress. And so there's something about the stress condition that triggers the overtaking of the alpha proteobacteria. All right, this is the chart that shows the ability of, when you get a sequence, sometimes you cannot identify them at the species level. Like if the sequence is really short from Illumina platform, you can only identify them down to the family or the genus level, but not to the species level. This is the proportion of the sequences that can be identified at the species level. And when they are full length, you almost guarantee, it's almost guaranteed that you can identify them at the species level. When they're not full length, sometimes you can only identify them, only 30% of them at the species level. And so if you would like to get a data that species identification is important for you, full length 16S sequencing would be much better than the short read sequencing. Okay, so if you would like to read more about how we develop the technique for full-length 16S sequencing with PacBio, you could look into this paper. All right, besides using the 16S or the amplicon sequencing, full-length amplicon sequencing technology for studying corals, we also use it for generating a DNA barcode for species identification for Thai herbals. And so the problem that was handed to us, oh, before we get into a problem, I would like to show you these videos. And so, you know, like how herbs are good for human, but they also very good for cat as well. So this is my cat. He's kind of weird because my mom loves to eat this butterfly pea flower for like antioxidants, and she started like feeding it to the cat, and the cat really enjoys it for some reason. He's like the weirdest cat ever. And you know, after a while, after you started feeding the cat with this for like a year, what you got is actually a cat that has got blue eyes from this. Believe it or not. Okay, I'm joking. So he already got blue eye. He was born like this. Okay, don't try to feed your cat with this and expect that it's gonna turn, the eye's gonna turn blue. Anyway, so the problem when we started this project, the problem that that was handed to us was that there was a lot of Thai herbs sold in the market, and this is what they look like when they're sold. It's in a capsule and nobody knows what's inside. You know, like for all you know, this could be grass. It might not be what they said it is. And so we need some way of identifying what species is actually packaged into the capsule. And the way to do it is that you can identify the species using the ITS, the RBCL, the PSBA, and MATK. These three are chloroplast genes that can do species identification in plants. But the problem is we don't have a lot of database. We haven't sequenced enough of the Thai herbal species. And so the first thing we have to do is to generate a database of herbal species in Thailand. And so we use a very similar technique to amplify PCR product from these genes and sequence them all using PacBio. And the trick we use, since we want to put 500 samples into one run in order to save cost, so PacBio suggested that you put the same barcode on the PCR product, right? What we did was that we decided to not do that. We wanted to do asymmetric barcoding, which means we put different barcodes on it. And so each combination represents one barcode. And if you do symmetric barcoding, if you want to pull or if you want to combine two different PCR products, you're going to need two different forward primers and two different reverse primers. Actually 200 difference and 200 forward and 200 reverse with the barcode. But if you do like a mix and match, you only need 15 forward and 15 reverse. And then you do the combination, you get 200 combinations. So it's a lot cheaper to synthesize 30 primers instead of 400 primers, right? So we did it this way and it actually worked, and it worked really well. And so we've been doing it since like this. And it's very similar to when I mentioned the 16S, except that now it's not 16S, it's RBCL, it's PSBA, and it's MATK. And you generate the library exactly the same way, you sequence it exactly the same way, and you process the data very similar. And you get the consensus sequence with very, very high accuracy. So just to sum up all the stuff that I've told you today, we have talked about how we apply next generation sequencing to identify SNP markers for marker-assisted selection, to develop elite varieties in rubber tree, in oil palm. Also, the SNP marker can be used for seed purity testing with cucumber, melon, chili pepper, also with corn and several other species. We can also apply a number of techniques to do biodiversity studies. We also use similar GBS technology to genotype 2,400 tigers in Thailand, because all tigers look the same. We don't know which one is which. So each tiger now has a DNA fingerprint. So if usually there's some people like smuggling tigers from Indonesia, from Burma, or from somewhere. And they always claim that, oh, this tiger is the kid of these two parents. And now we can actually do the DNA test and answer the question of whether a particular tiger has the other two tigers as parents or not, because this is like a big problem in the smuggling market. And now that we genotype all the 2,400 tigers in Thailand, we have the database. And then we also apply the PacBio sequencing technology to look at coral diversity as well as Thai herbal species identification. And so I'm not sure if anybody has any questions regarding any of the stuff in the lecture. I'll be happy to answer them.
So this is the last slide of your presentation, yes?
Yes, so it's a little bit
so
faster. I probably talk really fast today.
Oh, okay. So it's time for the questions if you have.
Yeah, if you have any questions.
I can see like some student like looking at your cat. So you said you can feed it with the butterfly pea and turn it to the blue eyes cat. Student like, what?
No, it's not true. Don't do that. Don't feed your cat with it. I always joke about that, but it's actually no, it's not true.
Okay. So yeah. So there's many interesting topics, I think, today. Are there any questions? Anything you can apply to your work? Many of the students are doing genome sequencing right now. You can ask Ajahn, how to approach to this and other when you get the sequence and other. There's many things. She's an expert.
We do a lot of genome sequencing as well.
Yeah.
Yeah, plant, bacteria, fungi, genome assembly. Yeah. But a lot of those are mostly.
What sort of question do you ask from your genome sequencing? What kind of question do you ask?
Usually when you want to know if the gene content, like if you want to know what annotated genes are, this is for bacteria and fungi. Like for bacteria, people are looking for resistant genes, AMR. For plant, we only do genome sequencing with the species that doesn't have any genomic resource, like loofah, like local species in Thailand that hasn't been sequenced before, and you want some like genomic resource for doing breeding or for doing some other type of experiment. Because without a genome sequence, it's very difficult to design primers. It's difficult to look for a gene. Yeah, but most of the genome sequencing project with fungi, I think typically either people do genome sequencing because they want to build a phylogenetic tree to look at like which clade or whether or not the two species are related. Yeah. I don't know what are you guys doing whole genome sequencing for?
Yeah. How about PP can tell her about your project? พี่ออย พี่แจน
Actually, I have like the project for whole genome sequencing for check the yeast evolution under the acetic acid tolerance, and like I want to compare between the unsettled strains like this.
So you like to look at SNP mutations over a period of time?
Actually yes, but like so the first thing I want to see the mutation point first, because like I just check the evolution just like to sample for compare for ancestral. Like actually I concerned about this also, because like we will have the data from the like two evo strain just to like is enough for to guarantee to SNP or not.
How many it depends on how many generations they are apart. Like the more DNA replication you have, the more chances that there will be mutations.
I'm not sure the replication, but like it take around like 6 months for evolution experiment. So I think like more than 100 replication of the yeast.
Then you might be able to pick up some mutations,
Yes.
if they're not deleterious.
Okay, so that's one project. What about พี่ออย
For me นะคะ did the ITS sequencing for finding the diversity of the yeast in the honey. And second one, I select some yeast that I isolate from the honey to whole genome sequencing, because I check the property of them. They can be produce some good flavor and aroma, and can be produce a lot of the acid. And for some species that I selected, they have some property like the probiotic property. And reason why I did it to whole genome sequencing to finding the genetic of them, and to deeply to study to produce a lot of the product of them.
Very good.
อาจารย์คะ yes, actually I have like wondering some slide like อาจารย์ show the F1 that caused from the parent, right? And then like how many sample to check for the SNP, or like conclusion to this one is the marker like this.
A lot. The more the better. Hundreds minimum. Yeah. So the more progenies you have, the more. So each progeny you only get. So you know, for each progeny you get some recombination events. So you would like to get a lot of recombination events, so you can look at where the break is, the break point. If you want to look at linkage, and if you want to get really close to the gene, you need a lot of recombinations. Yeah. So you could do it with 100, but that will give you a really crude map. But if you have 1,000, then your map would be like high resolution. Yeah. With yeast, you could probably do 500. With plant, it's a lot. You know, with oil palm, it takes a lot of land, a lot of fertilizer. It's difficult to grow 500 palms, so they grow 100. But with yeast, 500 colonies are easy to get.
Mhm. Yeah. How about the first year student? Any questions? Can you follow today?
Yeah,
We also
it might be
have
harder for
master and
first year.
PhD together. So how's the master student doing? Are you okay? Okay, Kap. Because you also have assignment for today too, right? So, yes. So maybe you can summarize. Ajahn present many sections on many interesting topics. So maybe you can select at least one section that you find is very interesting to summarize this.
Not the cat eating the flower section.
Yes. Okay. Any more question? Last question. How about we turn on the camera and take a group photo together, please. Before. So, and everyone, please turn on your camera. Who's the host?
พี่โบค่ะอาจารย์
Pibo. Okay. Pibo. Where is Pibo?
โอ๊ตน่าจะเดินไปเข้าห้องน้ำเมื่อ
ห้
กี้
อง
นี้
น้ำค่ะ she go to the toilet ค่ะ
Can someone help? I think her computer is probably in the room. And I think there's some people from somewhere also. Could you please introduce yourself, if possible? Aida, Uma, do I pronounce your name correctly? Fauchil, Udrick, are you there?
Okay.
Hi. Hi. Could you introduce yourself?
Okay, thank you. My name is Majarati, and currently I'm doing research in genetic cell, so I'm interested to join this webinar. Thank you so much for the interesting presentation.
So you doing your study where?
In Mahidol University.
Mahidol University. Ah, how do you hear about this seminar?
From my friends.
From your friends.
He is study in community.
Ah, okay, okay. Okay, welcome to join us.
Thank
so
you
is
so
Bo
much.
there?
Yeah. มาเร็วถ่ายรูปกัน
มาถ่ายรูปกัน โอเค so everyone please smile
ใครถ่ายอ่ะ
น้องโบถ่าย
ใครถ่ายอ่ะ
เป็นโพสไม่ใช่เหรอ โอเค one more time
เดี๋ยวกลับมาตรงนี้
โอเค ready
Yeah, not yet.
ให้โบเป็นคนเขาแล้วกัน
สักครู่นะคะวิตามินโอเคand 1 2 3 and capture กดตรงนี้ Okay, I'm ready everyone. Okay, 1 2 3. One more please. Capture. Okay. 1, 2, 3. Okay.
Okay.
Okay, thank you, Ka. So, lastly, I would like to thank Dr. Wiranda,นะคะ, Head of National Omics Center, that's kindly give a talk today, นะคะ. So, thank you very much.
You're welcome.
Yeah, okay, Ka. And I will keep in touch with you after this
Okay.
class.
All right.
Okay.
Thank you
Everyone
very
say
much.
thank you.
Thank you,
Bye-bye.
ค่ะอาจารย์
thank you ค่ะ
Bye-bye. Thank you for joining the class.
อ่า
อ้าวมันมาครึ่งเดียวเนี่ย stop recording แล้วก็